Mutation. It is the key to our evolution. It has enabled us to evolve from a single-celled organism into the dominant species on the planet. This process is slow, normally taking thousands and thousands of years. But every few hundred millennia, evolution leaps forward.
A, C, G, and T, adenine, cytosine, guanine, and thymine. These are the four chemical building blocks of the DNA molecule. They are components of nucleic acid, which gives DNA its tongue-twisting full name, deoxyribonucleic acid. The DNA molecule is made up of very long chains of the four bases, A, C, G, and T. In 1950, Irwin Chargaff published a paper stating that in DNA of any given species, the ratio of adenine to thymine is equal, as is the ratio of cytosine to guanine. This is known as Chargaff's ratios, and it was a crucial clue that helped solve the structure of DNA. Chargaff's ratios are universal. All forms of life obey this rule. Only the balance of AT pairs and CG pairs varies between species.
The Cavendish shop was to build us some tin models, and that took too long. And, finally in desperation, I made some out of cardboard. I began moving them around, and I wanted an arrangement, where I had a big and a small molecule, and so how did you do it? Somehow you had to form link bonds. So here's A, and here's T, and I wanted this hydrogen to point directly at this nitrogen. So I had something like this. Oh. So then I went to the pair, and I wanted this nitrogen to point to this one. And I went like this. Whoa, they look the same. And you can put one right on top of the other. We knew we could just, even if we go up to the ceiling, we were building a tiny fraction of a molecule. 100 million of these base pairs in one molecule, all fitting into this wonderful symmetry which we saw, the morning of February 28th, 1953.
The structure of DNA that Watson and Crick discovered suggests how genetic information is passed on. Before a cell divides, the double helix unwinds and the two strands of the DNA molecule in the nucleus separate. Each strand is then used as a template for the construction of new DNA molecules. The replication of DNA is simple in theory, but much more complicated in reality. The precise details have only recently been worked out. Take a look at the animation called replication mechanism to see what actually happens at the molecular level. This is Linus Pauling's failed attempt to predict the structure of DNA. The problem with his triple helix model is that the phosphates form the helical core, with the bases pointing outwards. This would be impossible under normal cellular conditions. Each phosphate group is negatively charged, and so many negative charges forced together would repel each other, literally driving the structure apart. The DNA double helix contains two sequences of nucleotide code letters that run along the molecule. Untwist the spiral, and DNA can be seen as two parallel strands. That's why it's sometimes called the double helix. Unzip the strands, and you have two linear sequences of the letters A, C, G, and T. The precise order of the letters along the molecule carries the coded instructions. These are the equivalent of binary ones and zeros in a computer. One strand is a complementary image of the other, and A will always pair with T, and C with G. So if you know the sequence of one strand, you can work out the sequence of the other. It dawned on Watson and Crick that this complementary copy of the code was the key to understanding how genetic information could be passed on. So the essence of life could be explained with chemistry. There were no mysterious life forces. Think of how many letters fit on a single page. And now imagine a stack of pages 300 feet tall. That's how much information is stored in the DNA inside every human cell, the entire human genome. If you sort through the 3 billion letters that make up the human genome, you find some surprising things. Only about 1% of the 3 billion letters directly codes for proteins. Of the rest, about 25% make up genes and their regulatory elements. The function of the remaining letters is still unclear. Some of it may be redundant information left over from our evolutionary past. The millions of bases which make up the human genome are organized into structures called chromosomes. These are arranged into 22 matching pairs plus one pair of sex chromosomes consisting of 2 X's in women and an X and a Y in men. So humans have a total of 46 chromosomes in each cell, known collectively as a karyotype. This set of chromosomes has a Y, so it must belong to a male. Only one chromosome in each pair is shown here. If you would like to see how our DNA is intricately folded up into chromosomes, view the animation called How DNA is Packaged. In this animation, we'll see the remarkable way our DNA is tightly packed up to fit into the nucleus of every cell. The process starts with assembly of a nucleosome, which is formed when 8 separate histone protein subunits attach to the DNA molecule. The combined tight loop of DNA and protein is the nucleosome. Multiple nucleosomes are coiled together, and these then stack on top of each other. The end result is a fiber of packed nucleosomes known as chromatin. This fiber, which at this point is condensed to a thickness of 30 nanometers, is then looped and further packaged using other proteins which are not shown here. This remarkable multiple folding allows 6 feet of DNA to fit into the nucleus of each cell in our body, an object so small that 10,000 nuclei could fit on the tip of a needle. The end result is that the DNA is tightly packed into the familiar structures we can see through a microscope, chromosomes. It is important to realize that chromosomes are not always present. They form only when cells are dividing. At other times, as we can see here at the end of cell division, our DNA becomes less highly organized. During DNA replication, both strands of the double helix act as templates for the formation of new DNA molecules. Copying occurs at a localized region called the replication fork, which is a Y-shaped structure where new DNA strands are synthesized by a multi-enzyme complex. Here, the DNA to be copied enters the complex from the left. One new strand is leaving at the top of frame, and the other new strand is leaving at the bottom. The first step in DNA replication is the separation of the 2 strands by an enzyme called helicase. This spins the incoming DNA to unravel it at 10,000 RPM in the case of bacterial systems. The separated strands are called 3' and 5', distinguished by the direction in which their component nucleotides join up. The 3' DNA strand, also known as the leading strand, is diverted to a DNA polymerase and is used as a continuous template for the synthesis of the first daughter DNA helix. The other half of the DNA double helix, known as the lagging strand, has the opposite 3' to 5' orientation and consequently requires a more complicated copying mechanism. As it emerges from the helicase, the lagging strand is organized into sections called Okazaki fragments. These are then presented to a second DNA polymerase enzyme in the preferred 5' to 3' orientation. These sections are then effectively synthesized backwards. When the copying is complete, the finished section is released and the next loop is drawn back for replication. Intricate as this mechanism appears, numerous components have been deliberately left out to avoid complete confusion. The exposed strands of single DNA are covered by protective binding proteins, and in some systems, multiple Okazaki fragments may be present. The molecular reality is very different from the iconic image of the double helix neatly separating into 2 DNA copies, as so often depicted. Using computer animation based on molecular research, we are now able to see how DNA is actually copied in living cells. You are looking at an assembly line of amazing miniature biochemical machines that are pulling apart the DNA double helix and cranking out a copy of each strand. The DNA to be copied enters the production line from bottom left. The whirling blue molecular machine is called helicase. It spins the DNA as fast as a jet engine as it unwinds the double helix into 2 strands. One strand is copied continuously, and can be seen spooling off to the right. Things are not so simple for the other strand because it must be copied backwards. It is drawn out repeatedly in loops and copied one section at a time. The end result is 2 new DNA molecules. The central dogma of molecular biology, DNA makes RNA, makes protein. Here the process begins. Transcription factors assemble at a specific promoter region along the DNA. The length of DNA following the promoter is a gene, and it contains the recipe for a protein. A mediator protein complex arrives carrying the enzyme RNA polymerase. It maneuvers the RNA polymerase into place, inserting it with the help of other factors between the strands of the DNA double helix. The assembled collection of all these factors is referred to as the transcription initiation complex, and now it is ready to be activated. The initiation complex requires contact with activator proteins, which bind to specific sequences of DNA known as enhancer regions. These regions may be thousands of base pairs distant from the start of the gene. Contact between the activator proteins and the initiation complex releases the copying mechanism. The RNA polymerase unzips a small portion of the DNA helix, exposing the bases on each strand. Only one of the strands is copied. It acts as a template for the synthesis of an RNA molecule, which is assembled one subunit at a time by matching the DNA letter code on the template strand. The subunits can be seen here entering the enzyme through its intake hole, and they are joined together to form the long messenger RNA chain snaking out of the top. Once a gene has been located and transcribed into mRNA, it must first be edited before it can be translated into a protein. This editing process is called splicing. It involves removing non-coding regions called introns, leaving only the protein-coding exons. In the cell, the introns are removed by special enzymes which recognize specific sequences. These enzymes cut and rejoin the coding regions for translation into protein. The job of this mRNA is to carry the gene's message from the DNA out of the nucleus to a ribosome for production of the particular protein that this gene codes for. There can be several million ribosomes in a typical eukaryotic cell. These complex catalytic machines use the mRNA copy of the genetic information to assemble amino acid building blocks into the three-dimensional proteins that are essential for life. Let's see how it works. The ribosome is composed of one large and one small subunit that assemble around the messenger RNA, which then passes through the ribosome like a computer tape. The amino acid building blocks, that's the small glowing red molecules, are carried into the ribosome attached to specific transfer RNAs. That's the larger green molecules, also referred to as tRNA. The small subunit of the ribosome positions the mRNA so that it can be read in groups of 3 letters known as a codon. Each codon on the mRNA matches a corresponding anticodon on the base of a transfer RNA molecule. The larger subunit of the ribosome removes each amino acid and joins it onto the growing protein chain. As the mRNA is ratcheted through the ribosome, the mRNA sequence is translated into an amino acid sequence. There are 3 locations inside the ribosome, designated the A site, the P site, and the E site. The addition of each amino acid is a 3-step cycle. First, the tRNA enters the ribosome at the A site and is tested for a codon-anticodon match with the mRNA. Next, provided there is a correct match, the tRNA is shifted to the P site, and the amino acid it carries is added to the end of the amino acid chain. The mRNA is also ratcheted on 3 nucleotides, or 1 codon. Thirdly, the spent tRNA is moved to the E site and then ejected from the ribosome to be recycled. As the protein synthesis proceeds, the finished chain emerges from the ribosome. It folds up into a precise shape determined by the exact order of amino acids. Thus, the central dogma explains how the 4-letter DNA code is, quite literally, turned into flesh and blood. Once the DNA double helix had been discovered, the next big challenge was to work out how the 4 letters of DNA could code for each of the 20 amino acids that make protein. The first question was, how many DNA letters coded for each amino acid? If it was 1 DNA letter for 1 amino acid, then you could only code for a maximum of 4 amino acids. 2 letters in every possible combination could code for up to 16 amino acids. Still not enough. But 3 DNA letters provide more than enough combinations to code for all 20 amino acids. So 3 was the answer. It was a triplet code. DNA is under constant attack from reactive chemicals and natural background radiation. Free radicals are the byproducts of normal metabolism in human cells. Seen here as bright particles, they sometimes react with DNA and cause chemical changes. Radiation can also affect DNA. For example, ultraviolet light from the sun can cause harmful chemical changes in the DNA of skin. These changes can lead to kinks in the DNA that prevent genes from being correctly read, or deletions that alter the type of protein produced. Thanks to constant biochemical repair work, most mutations are corrected before they have any effect. But in rare cases, mutations can accumulate, and this can give rise to diseases such as cancer. Sickle cell anemia is a genetic disease that affects hemoglobin, the oxygen transport molecule in the blood. The disease gets its name from the shape of the red blood cells under certain conditions. Some red blood cells become sickle-shaped, and these elongated cells get stuck in small blood vessels so that parts of the body don't get the oxygen they need. Sickle cell anemia is caused by a single code letter change in the DNA. This in turn alters one of the amino acids in the hemoglobin protein. Valine sits in the position where glutamic acid should be. The valine makes the hemoglobin molecules stick together, forming long fibers that distort the shape of the red blood cells, and this brings on an attack. Polymerase chain reaction, or PCR, uses repeated cycles of heating and cooling to make many copies of a specific region of DNA. First, the temperature is raised to near boiling, causing the double-stranded DNA to separate or denature into single strands. When the temperature is decreased, short DNA sequences known as primers bind or anneal to complementary matches on the target DNA sequence. The primers bracket the target sequence to be copied. At a slightly higher temperature, the enzyme Taq polymerase, shown here in blue, binds to the primed sequences and adds nucleotides to extend the second strand. This completes the first cycle. In subsequent cycles, the process of denaturing, annealing, and extending are repeated to make additional DNA copies. After 3 cycles, the target sequence defined by the primers begins to accumulate. After 30 cycles, as many as 1 billion copies of the target sequence are produced from a single starting molecule. A common technique in genetic engineering is to insert a new gene into a loop of bacterial DNA called a plasmid. The molecular tool used to cut DNA is a restriction enzyme such as EcoR1. The enzyme has a precise shape that allows it to run along the groove of the double helix, scanning, in the case of EcoR1, for the base letter sequence GAATTC. The enzyme then cuts the plasmid at this specific point, allowing a new piece of DNA to be inserted. When it cuts, EcoR1 leaves a sticky end. This helps the new gene to attach. The joins are then stitched together by another enzyme called DNA ligase. The genetically engineered bacteria is grown in a culture medium. Very quickly, large numbers of the bacteria can be produced, each with a copy of the inserted gene. The bacteria duly manufacture whatever protein the gene codes for, and so the desired product is made. The first method of sequencing the genetic code was devised by Fred Sanger. To sequence the DNA, it must first be separated into 2 strands. The strand to be sequenced is copied using chemically altered bases. These altered bases cause the copying process to stop each time one particular letter is incorporated into the growing DNA chain. This process is carried out for all 4 bases, and then the fragments are put together like a jigsaw to reveal the sequence of the original piece of DNA. As represented by this huge stack of paper, the human genome contains more than 3 billion nucleotides, or DNA letters. The first stage of the public Human Genome Project focused on identifying marker sequences, or unique tags, shown here in yellow, at regular intervals throughout this Book of Life. Once enough sequences were tagged, various blocks of the genome were allocated to different academic centers for sequencing. To begin the sequencing process, several copies of a section of DNA, represented here as a page of text, are cleaved to produce smaller fragments. Although it looks fairly orderly, this step is small-scale shotgun, which creates numerous random fragments. Each fragment is sequenced. Then computer programs align the overlap between fragments to build up an entire page. Marker sequences, shown in yellow, help establish the order of pages in the Book of Life. This methodical process produced huge amounts of data that have been used to virtually reassemble our genome. However, there are gaps. Repeat sequences are common in the human genome, so repeats from entirely different chromosome regions may be erroneously joined together. It will take many years to detect mismatches caused by the repeat sequences. Some regions, especially near the centromeres, may never be fully finished.
It's an example where human embryonic stem cells are grown in a culture dish, and we remove the factors that allow them to self-renew, and they now spontaneously differentiate. Could I have the next video, please? And what you'll see here is that these cells, in some cases, spontaneously make beating muscle. cardiocytes, that is the muscle just like in your heart. Now, of course, here we're seeing 4 examples of that. You see that they beat at slightly different rates, and obviously they're not organized into anything like your heart. Your heart, which Nadia will be talking about, is about the size of your fist. This is a tiny little group of cells, thousands of cells in a petri dish. But it does raise the interesting question of how did these cells know what to do? How did they make this decision to become cardiomyocytes? What steps were involved? What were the signals that they received perhaps from their neighbors? What I'd like to leave you with then today is this fact that embryonic stem cells can make all cell types. How do they do that? We don't know how they do that, but it's an exciting problem because it'll teach us something about normal development, and it also has the potential to treat diseases where cells are missing, and I'll be talking more about that tomorrow. This video is prepared by my colleagues Dieter Egli and Kevin Egan, and you see here on the left is what's called a holding pipette, which gently sucks on the egg. The egg is surrounded by a membrane called the zona, and you'll watch this pipette on the right first drill a hole into the zona, then go in and suck the nucleus out, and then another nucleus, which has been taken from, say, a somatic cell, a cell of the body, is going to be put in. So if we could start the video, please. You'll see now that this drilling pipette is going to drill a little hole into the membrane. You can maybe see a little bit of the hole right here at the next part. This pipette, you'll note, isn't really sharp like a syringe. There you can see a bit of the hole. And now the pipette's going to go in and remove the nucleus. And if you look carefully in the pipette, you'll see a line in the nucleus, which are all the chromosomes lined up. So that nucleus is going to be squirted out now because we don't need it anymore. And there we have an enucleated egg. Now the next step is to take a set of eggs like that, and I'll show you 2, and then transfer into them a nucleus from another kind of cell, a fully differentiated somatic cell. So here the enucleated egg is set on the side, that it's held by this holding pipette on the left. There's drilling the little hole in the membrane. Here we go in. Here comes the nucleus from the right. The pipette goes in, and these pipettes are operated with a piezoelectric device. So you can't see it here, but it's like a little jackhammer going very quickly, like a Woody the Woodpecker, getting in there to then squirt the nucleus in. Here we'll see it again, a little hole, and the zona is prepared, and now the nucleus is going to be squirted inside. So there's two examples of what's called somatic cell nuclear transfer.